eyes on the left side, and so that's informative.
Thank you, Chris. I have to say, watching this guy move is just fascinating.
Yep.
One of the things that's been great about all of these dives is seeing this diversity of fishes and how they adapt to their environments.
That's pretty fine movement.
This guy's just weird looking. You get the feeling he's slowly but surely trying to get away from us. Not very quickly.
Okay. Okay, video is clear.
A little feather in the back. All right.
All right, are we ready to move on? Thank you.
Forward.
Thanks.
Oh, that's a very good point. So in our event log, Dave Burdick, who's at the University of Guam, says it looks like it moves like a polychaete, and it totally does. It actually moved very much like we expect these scale worms to move.
Maybe not. Never mind.
This pile of stuff.
I thought I saw one that might have been.
So I think we're still in an area of mostly volcaniclastics.
Oh, you got a bunch of urchins again up top.
Yeah, right to your port corner.
What's that?
If you look, you got a bunch of urchins on that big rock right there.
Oh yeah. Good eyeball on that. Sometime here. So are you lined up on our current track, or is it
about 2.5? Well, I'm going back like 5 degrees both ways, but 2.5 seems to be pretty good.
Okay.
You got some real nice serious cams picking up some big stuff to the starboard ahead.
Cool. Go take a look.
So we're seeing more of those same sea urchins that we saw at the beginning of the dive.
So this is a little rubble pile. Does any of this look loose to you?
It does look loose.
Do you want to poke, or do you want to just keep heading on?
Hold on just 1 second.
Okay, I'm going to stop the ship if it hasn't already.
Chris, would this be
You want to give it a try for a rock here, maybe?
I absolutely would. Are you okay with that?
Yeah, absolutely. I've actually been trying to look and see if I could spot one that looked loose.
I think this looks like a good area, so I think we are going to try to stop. Patty, does this look good to you?
How far do we got left in this ship move?
We've arrived at our new position.
Copy. Thank you.
Oh, good. Patty said yes. And Bob, I'm going to assume that you're going to like this rock.
Yeah,
You're right.
the ones under the ledge
Yep.
look particularly promising.
I agree.
Look like they're glued to that brown, whatever it is.
Yeah, so Pilot, we think that one sort of where you're, I think, moving towards right under that ledge is probably a good place to hunt.
Okay. We'll give it a try. See if we can find.
Boy, I don't know. When he spins around a little bit, they look like they're just as glued as the other ones.
Yeah, well, we'll try it, and if not, we'll just keep moving.
If they look glued, maybe we don't want to use the time.
I mean, some of them look like they might be loose. It's hard to tell.
Right of the sea urchins, you can see a rounded thing that looks like a lava tube, you know. It looks certainly like
I'll sit here
the
for a second.
fractures in a typical pillow fragment, and you can also see jointing fractures on the ledge that's right above the little underhang.
Right.
Okay on
That's all
sonar.
out of view for us right now, but it actually might be on the quad screen on stream 3.
I'll keep it on my camera. I'm at 75 right now. If I have to adjust it
Let's
more.
give it a try and see if we can grab one of these rocks.
Yeah, ship's definitely not moving.
I'll reach in there as far as I can.
I'm
pinned up almost. I can't pin against the wall, unfortunately, because of these coral and stuff.
watch
I don't
your
want to squish them. But
Okay.
arm
But
when you come out.
I am gonna.
So if none of these on the right budge, might be worth a shot at the rock that's directly under the coral fan.
Oh, the one that's a good idea. I agree, Chris.
Say again.
Pilot, what about the one that's under the coral fan? So the black one that actually looks like it's not necessarily sitting in a sediment pile?
Oh, way over on the left.
Yeah, can you reach that, or is it too far away?
not the way I'm settled now. I can't reach it,
Well,
but
then
I can
let's,
get into a configuration to
before
try to
you
reach
do
it.
that, why don't we try the ones that you can reach?
Okay. We'll poke around here first. Copy that.
So we use these geologic samples to try to characterize the site of the area. We can use them for age dating. We can use them for geochemical composition. They're helpful in order to find out really what the history and formation of these areas are.
And if I could add,
Please do.
we're interested in the processes that are associated with subduction zones. This is an island arc above the subduction zone, and from the composition of these lavas, we can infer a lot about the processes in the subduction zone.
Great. Thank you, Bob. That's totally true.
Yep. Well,
Those guys are set in their ways, aren't they?
Yep, they are.
Just when we think we see promising rubble.
No, that guy's not close. Okay, we can go for the one Chris was suggesting, or continue on. Up to you.
Starting to get somewhat.
How long do you think we'd try to set up for that one last grab underneath the coral?
A minute. Maybe.
Okay, then please let's do it.
Okay. Get over there real quick. Looks kind of big from this angle, doesn't it? Right, we'll poke around in this pile. There's a couple that might be loose here. Work. Bonk. Alright.
So is that another urchin down there
Yeah.
in the
different
crevice?
Type of urchin, maybe?
Yeah, that's what I was wondering, if it was the same urchin or a different urchin.
Looks like a different
He
kind.
wasn't invited to the party.
That's a different urchin, I think, and I think it's in the family Sideridae, pencil urchin.
Oh, thank you, Chris.
Copy, Nav. Bridge's move completed.
Copy. Thank you.
Totally stable, but pretty close. I'll try this real quick. Alright, we'll try this guy real quick. Nope, also not loose. Fortunately. Once again, we were fooled.
Okay. All right, so we are not going to be successful at getting a rock sample here, but there is a fish that's hanging out. Actually, several. So I think we can just keep moving, because these are not the rocks for us today.
Copy that.
Thank you, pilot.
You're welcome.
All right, so we will continue our hunt upslope.
Yeah, so just ahead, it looks like there's a...
We are at a depth of
A lot of
490
things
meters. Oh, sorry, Chris, go ahead.
Possibly.
Sabling valve packs.
Thank you.
I would really love to know
Huge
what
saving
is that cement that's gluing those
So yeah,
structure fragments
good delta
so
on
well
this 20,
together.
so you
This
can
is
pick up and
really quite
move ahead,
unusual.
no issues.
I don't disagree. I don't think it's possible if we can get a loose
Pilot, what's
piece,
your
we might
max
be able to
speed
get some
on
of the
V2?
sediment as well, which has happened
Like
multiple
forward?
times over this expedition.
If you know, I've seen little ledges of that.
We need a
cement-
deep sea rock
cemented,
drill.
Sediment, sort of hanging off
Mhm.
on certain spots. If we see that, we do have another arm that we're willing to squeeze a little harder with, that we could try pinching a little piece off and then picking it up. It takes a little more time, so it's a time trade-off. So it's up to you if you want to spend the time doing it. It's
Yeah,
really worth it.
I'm
But,
not sure about that, but
um,
thank you.
But it is like see like that, so that kind of area where it's sort of
Yeah.
laid over or whatever. But just food for thought.
Okay.
not pushing forward or anything.
Thank you.
Obviously. And we have a big field of biology coming.
So you guys have to look in the Sirius camera right now. So check out stream 2. You just have to see this. It's amazing. I expect that all of you are switching over to Sirius
we're
right
talking.
now. Look at that.
Yeah, now
Look
we're
at that.
talking. That is amazing. I wasn't looking at that.
I know. I was like, you got to go check it out. So, and I realize that normally I'm the one talking about geology, but that's just amazing. There's so much biology that's coming up. As we move up, Chris, please tell us, or Scott, tell us where we should be zooming in, or anyone.
Right. Actually, it looks off to the side of the cliff, too. There's a lot of life.
Yeah.
Okay, well, first of all, you have some tubular sponges that are upper center right now.
Do you want to zoom on those?
And we have not seen these before. I think I know what they are.
Can we zoom on those tubular sponges to the left
This
center?
would definitely be worthwhile taking a look at.
We are doing so right now. They are so beautiful.
Well, that looks like most of what's here is these sponges.
Yeah, although there was something yellow to the bottom right as well that we might want to try to zoom in on afterwards.
Yellow bottom right now, you mean?
Yeah, right above your skid.
Yeah, right in front
Like just one out of frame.
Right toe.
But let's start with these tubular sponges.
Copy that. Tubular sponges first.
Doesn't that sound like it should be a band name? Tubular sponge. Only me, huh? Okay.
No.
Okay, so I'm pretty sure I know actually what species these are, but I'm going to wait for the close-up. I've seen them once before, and I think it was on a dive off of Palmyra that I was annotating.
Still settling.
Zooming in.
Oh, these are cool looking.
Holding. Partial.
Those are real cool.
All right, so we're zooming in on these tubular sponges. We've got the lasers on, so you can see the 10 centimeters. And you can see a squat lobster hanging out in the background. And Chris, you want to give us your most educated guess? Oh, and what's that at the bottom? Is that a fish?
There is a fish at the bottom.
That is a fish.
So Chris, is this a Silocalyx wilsoni? And a scorpionfish.
Yeah, as you can see, it's a really distinctive sponge.
Absolutely.
So I don't even remember what the family is, but somehow the species name just popped into my head. I think because I really liked them the first time I saw them. Unfortunately, they don't make a good collection if we think we can actually go to species name here.
Okay, I think we can move on for another zoom when you have when you're good.
Do you want the darker ones? Is that better for your light?
Actually, I think the brighter ones
Oh,
are
look,
better.
another little squat lobster.
Better, okay.
Yeah.
There you go.
Thank you.
And when video is done, we're good to move on to something else.
Roger.
Thank you.
Okay, video's clear.
Pilot's clear. Yep.
Full white.
Copy that.
So I think they
Just a
went
little
with
bit
the
different.
yellow to the right.
Yeah, it's a little bit different. That's just a feather star.
So which way do they filter the water, from the inside out or the outside in?
I think the water's coming in the sides and then goes out through the top, the large osculum on top.
Do they just sit in the...
Chris, is there something else specific or biologists
right here.
in the
See if
event
this
log?
is
Is there
the
something
same or
specific you would like to
smaller
zoom on?
again, or is something different?
Well, we can zoom on what they're zooming on right now because this is probably something different. And yes, it is another type of sponge that I've seen in very poor video and couldn't go any further than sponge. So this is really interesting unless this is a very, very young form of the Silocalyx.
Zooming in.
Okay.
Holding. Full zoom.
Roger.
There was also, after this one, there was a leafy type of sponge that was to the right or starboard of the vehicle, and I think it's just more turning that way. I don't think it's too far behind us.
Okay.
And if we can't get it, that's fine. It looks like we're about to enter a huge target-rich environment.
We really are. There is so much coming up. I think it's going to be just like an embarrassment of riches in terms of being able to zoom around and look at fantastic stuff.
Okay.
This is great. Okay, video's clear.
Great.
Zooming out.
Leafy sponge. Leafy sponge.
Full wide.
Thanks. Just gonna back up and wag.
All right, so I think we wanted to pan to the right and look for a leafy type sponge.
Oh, I know which one he's talking about, possibly.
There it is, right at the bottom.
To the right of the tubular sponge?
Now.
It's right here, yeah.
Yep. All right, we're moving in on it. Chris, do you want to look at any of these corals as well that are on the side or the yellow thing that's on the edge of the screen? Just let me know. We can do that afterwards. There's also a fish in the bottom. It's probably the same scorpion fish. Something just flew by.
Start in on the leafy sponge.
Zooming in.
Yeah, I mean, the corals may be that Narella again, and I think we're going to see a lot of those upslopes, so we could just go up a little further for that one and get some more of those.
All right, so instead we're zooming on this other sponge. Chris, can you tell us what it is?
Well,
you're the one who wanted to look at it.
I can take a guess. I think it's in the family Eurydidae. It's a hard sponge and very, very brittle. I don't see very good tube development here. This is more sort of leafy, so about the best I can do is Eurydidae.
And it also has at least two squat lobsters, very little ones. Are those hydroids up in the top right-hand corner?
Yeah.
Or zoanthids? I'm not sure which. Anyone? Help?
Does have something growing in it, doesn't it?
It appears to. I just don't know what it is.
Could help with the ID.
So you can see them. Actually, they're throughout the whole area. You can see them easily on the top, but they're really throughout. When we just zoomed in, you could see them.
Can we tilt up?
Up, up, up.
Perfect.
Yeah, of course, the one day that I don't bring down this book about New Zealand sponges with me, we see something that would have been already documented in New Zealand. I remember reading something about a sponge that had zoanthids on it like this.
So these could potentially be either zoanthids, hydroids, or anemones? Is there one that is more likely than others?
I'm going to zoom out.
I don't think it's a hydroid, so I think it's either some type of anemone or a zoanthid.
Awesome.
Right there's fine. Zooming in, holding. That's nice.
Okay, when we're done zooming on this one, do we want to try to keep moving, or is there anything else you'd like to look at?
Well, why don't we get up into that main bed and take a look and see what we got? I think there'll be quite a lot of things.
Absolutely. And I will mention, for those of you who are also watching feed 3, I asked to have the left-hand upper switched so that you could kind of get a view of what we were moving towards upslope. So take a look at that and see if it's of interest to you as well. And for those of you who are just joining us now, we are at Esmeralda Bank, which is also North Esmeralda. I believe it's an extinct volcano as part of a larger volcanic complex. We are at a depth of 483 meters. We are moving up a very, very steep slope that appears to be volcanics and probably volcaniclastic sediment. We started out in a very exciting place where we were looking at tons and tons of urchins, and now we are in the midst of sponge and coral wonderland. And scientists, please let me know when you would like to zoom on something. Just feel like saying, Happy Mother's Day, we brought you a sponge. Ta-da! A bouquet of corals, perhaps? Is that a crinoid down on the side left?
Yeah.
You can keep going.
Yeah, that might be a good one to take a look at.
Okay, then we'd like to look at that, please. if you can look at that one or the one that's above it.
I'm not
There's
sure.
like 3 crinoids in view. So
Here, what?
Coming in on the bottom one.
perfect.
I haven't heard it for a minute or two, though. I don't know where it came from.
Start in video.
Zooming in. Holding.
Thanks.
So we've got multiple squat lobsters. We've got this crinoid that appears to be sitting on. Is that a sponge covered in something? Like the zoanthids or the anemones? Anyone?
Yeah, it sure is. They're so covered that you can't even see what the sponge is. It could be a farreid or another euretid.
Wow.
Am I going any closer?
Sure.
Okay.
Zooming in. Holding.
So we're just zooming in on this crinoid. And I'm assuming that these are not the stalked crinoids, but these are the other type of crinoid I can't remember the name of. A commensulate crinoid.
So you can see some of the sponge material right there in the center bottom, and it looks pretty similar to the one we just got an image of. And so my best guess would be it's another euretid.
Perfect, thank you. Could you put that in the event log? Because I can't spell it. All right. I think whenever video is ready, we're ready.
Just a few seconds here. That's a nice shot.
This has really been just a fascinating dive already today. We've seen so much.
Zooming out.
Thanks.
All right, and we're just going to keep moving along, stopping to zoom when we find something interesting. As you can see, we see both sponges and corals, a number of those crinoids. And every time we zoom in, we seem to see another squat lobster.
Oh, there's a cool urchin.
Clearly they're squatting.
You guys want to ship move a little one, a little jump?
Yeah.
We have some mat of something that's coming in the upper left corner that looks kind of interesting.
Would you like us to zoom on it?
Look at that.
Okay.
It looks like
Do
there's
you want
still
the
work
urchin
up.
or that?
Let's try the thing that's off in the left.
Off on the left, okay.
Go
Yeah,
by, it looks
I don't
like
know
you're
what that
still
is. I
lined
think
up
that's what
on
they're
the,
looking at.
This sort
Make
of
it
brownish.
three
Yeah, I
looking
mean,
stuff?
to me it looks like something
Starboard,
dead.
Yeah.
Oh,
Is what?
3015.
Can we just zoom in on that?
we're going for it.
And hopefully someone knew what kind of urchin that was that we passed by.
Bridge nav.
Fish is swimming backwards.
Can we get a ship move 15 meters to 15
Okay.
meters at
Come in.
030 Zoom in. degrees, 0.2 knots? Holding here. Yes, 030.
More of what we saw on the bottom earlier.
Yes.
Hey, this is
I
where
mean, is
came
it
from.
just old dead coral that
Looks like it to me.
it's and a fish?
So this is a type of scleractinian coral, and I think Dave Burdick is watching right now, and maybe he can sort of weigh in on this, if nothing more in the chat. This part's dead, but I'll bet there's some live sections of this coral somewhere in this huge clump anyway.
Keep scanning around for the live.
Yeah, please keep looking, and maybe we can find a part that is alive. I mean, so far it looks pretty dead to me.
That's an incredible concentration of coral.
So there's certain types of deep water
Oh, look, there's a gastropod.
sclerotinians that form really large mats, and looks like we've come across something like that. Oftentimes the underlying coral sort of dies off, and then right at the ends of it or the upper parts of it, you'll still see you have some live tissue.
Well, we are
Call this a coral reef?
No, I believe it's a coral mat.
I don't know if you have it a coral. I'm not sure you call it a coral reef. Right now, everything's dead. I haven't seen any live tissue yet. So Sonia Raleigh has thrown out a good guess, Somalis amelia, which is fairly widespread, I think, in the Atlantic, and also has been documented in the Pacific. We actually documented it in Hawaii, but only a huge dead graveyard of that coral. We've never actually documented the live Somalis amelia.
Yeah, a little bit more.
Are we getting any closer here? Because all of this looks pretty dead to me.
I see a couple polyps. Oh no, that's just overgrowth.
Yeah, it's pretty dead.
It's all white, not brown, but it doesn't seem.
Do we want to continue to explore in this area?
Can we go up and see if we can find any alive stuff?
Yeah, we can probably leave the dead stuff alone, and I think we can spot the live stuff pretty quickly as we're cruising up slope.
Okay, great.
All right.
So, pilot, whenever you have a chance, let's keep moving.
Thanks. Roger that.
It's really interesting how the sponges have just sort of taken over that area.
Yeah, there's all.
And it just continues upward. Wow. Is it worth tracing this path upward to see if we find where the live ones are?
You don't know. I mean, this might just be where they're collecting.
Yeah, that's true.
There's probably a ton up top, and this
all right,
is
let's
like a spot they collect. Maybe.
Let's keep going. Let's see what we can find.
Follow it, see where it goes.
Wow.
I've never seen so much dead coral in one spot before.
What's the guy that's moving? Going off screen.
What's that?
Nothing. Something was moving and it went off screen that we didn't see.
Ooh, is there any fish there?
There's a fish in the top center. Could you zoom in on that?
Yep.
Zooming in. Are you ready?
Yep.
Zooming in.
Seems like a coral landslide.
Take it a little further. If you want.
Zooming in. Holding.
Is this a type of roughy?
I think you're learning some biology, Deb.
I think I may have been looking at Brian Kennedy, who's sitting next to me in the back, who's feeding me answers. Although I did get the crinoid right on my own.
That's cheating a little bit.
It's totally cheating. It's not even a little. It's a lot. So are these commercially important species, or are these relatives of the commercially important, like orange roughy?
So I bet if Bruce is here, he'd probably say this is Hoplostethus crassuspinus, and I'm just leaving it as Hoplostethus.
Okay.
They're getting good video, so Bruce will be able to weigh in on it.
Great.
The commercially valuable species is Hoplostethus atlanticus, even though they're here in the Pacific. And I don't think this is it.
Okay, great.
I think these are much more infrequent, solitary kind of versions of that genus.
Awesome. Thank you.
That's nice, Jeff. Zoom out a bit.
Okay.
Holding.
He's really bright. Reflective guy. a lot of momentum to switch directions.
Yeah. Well, I think we're clear.
All right, thanks.
Zooming out.
All right, so there is so much to
Wide.
explore. If you look in the Sirius cam,
Look at that
it's kind of
jellyfish out.
amazing.
Yeah.
Just about, you know,
Floating
we could choose to go
jellyfish.
anywhere and see many, many things. I think we want to
Try the
continue
old jelly
upslope
or-
after we're
Oh, we
done
can?
imaging
Sure.
this fish.
All right, going out to it.
Zooming in.
Yep.
Zooming in. Oops. Holding.
Oh, that's not a fish. That's a jellyfish.
Yeah. Bring him out. There we are. Yeah, he's real close.
Is he?
Zooming in. Or zooming out.
Oh,
sorry.
do we lose him?
Yeah, I can't keep focus.
Yeah, I'm gonna
Nope.
pull him away a little bit. Backing up. He came right into me.
Zooming in.
Oops.
And holding.
Might be too hard to get him. There was a glimpse. Maybe someone could get a snap from it.
Yeah, alright. Zooming out. Full wide.
Couple of them were those urchins. Perched. Continuing on. Now you've changed all your settings for that.
Yeah. Get it straightened out again here.
All right, so we're continuing to move up this slope.
This is a little different here. Maybe.
We're still seeing a lot of these very tubular sponges interspersed with coral. Scientists, tell me where you'd like to zoom, and we will make that happen for you.
Let's see if this thing is of interest. Want to start in?
And we want to try to keep an
Oh,
eye
we have
out.
a colony that's coming to the center right now that might be different.
Holding.
All the ophiurids on it. It's upper left right now, right at the top.
Oh yeah, right there. Can we zoom in on this, or
We are zoomed in on it
can we get a little closer?
shortly.
The ship has stopped.
Thanks.
Copy nav.
Okay. You can get a little more if you'd like. But
Zooming in.
Try to hold it.
Holding.
And we will try to continue as we move up slope, looking for that live scleractinian coral. We just saw extensive dead mats of the scleractinian. So can someone tell me what kind of coral we're looking at?
Yeah, so my best guess is it's a primnoid, and it's maybe in the genus Phenelia. I think it's either Phenelia or Caligorgia, and my first choice would probably be Phenelia.
We want a tighter shot.
Great,
Get a
thank you.
foothold.
I can zoom in a
And
bit
so they're
more.
it's just totally covered in these ophiuroids.
Full zoom.
And we know that these ophiuroids like to climb up into the coral so they can get a little further out and have a more activity for their suspension feeding.
All right.
That's fine, I think.
Thanks.
Zooming out. Full wide.
I lost with the a little bit of currents tending. I think it's pushing me now. South.
Can we zoom up into the top left corner?
To Georgia. Possibly.
Good spot there, Deb. That looks pretty interesting, that purple one, right?
Thanks, yeah. And I think there may have been a sea star or Brazingid at the bottom that we just passed.
Yeah, I was going to get the little orange guy before the purple guy, but
That's okay, we like this purple thing.
didn't seem like the orange was of interest. Going for purple. I see a spongeless rock I can plant for this guy, so that'd be good.
Pilot, do you care if we put another move in? 15 meters? Alright, Nav, do you want to get another move in? I think same
Still
thing.
like the same heading?
Yeah, 30, 15 meters.
Copy. Bridge nav.
Okeanos. Can start in video. Hold this.
Zooming in.
Hold.
Hey, we're going to put a ship move in, same as before. That's 15
Holding.
meters, 30 degrees, 0.2 knots. Good copy.
Zooming in.
So what is this
Holding.
purple thing we're looking at?
Zooming in.
Anyone?
Holding.
I think
Zoom.
the coral biologists are still collating.
Wow, look at that.
So all the polyps are closed up here, which makes it a little harder to figure it out.
So one guess from Sonia Rowley is that it's a Victigorgia, which we have seen on previous dives before, and it does have this very distinctive purple color. Oh yeah, what is that?
I'm just going to scan along a little here.
Sure. Yeah, mid full zoom. So
Interestingly enough, the coral biologists are saying that Victigorgia is actually probably the only purple sponge that they know. So that's the guess, because it's purple.
Oh, my other turns.
Well, one thing, if you take a look at the closed up, the tentacles, you can count 8, so it's going to be an octocoral. And it looks like it's not growing on something that's dead. It looks like that stem really belongs to it. And so I think we all kind of know of only one right now that would look like that and do that, and that would be Victigorgia.
Great. All right. I think we can move on when we're ready.
yeah, video's clear.
Okay.
Zooming out.
Thank you for the zoom. That was fantastic.
You're welcome.
Full wide.
I just wanted to make a comment that right now we're not in the volcanic clastics anymore. We're in massive basalt.
Cool. Thanks, Bob. Where do you think we went through that transition?
By the way, the cavalry has arrived. Scott France is back online, so now we've got a nice group of people who can figure out what some of these things are.
All right, we've got no less than like
Wait,
So
4
it seems
coral
like
biologists.
about 3.5
It seems
now.
like the sponges are like this dense basalt. They don't like the volcanic clastics.
Yeah, it sure looks like that, Bob, doesn't it? And I think the corals like it too.
Yep.
So right now I'm at a 4.2. That's just me being blown off, I guess, by current.
Oh, what kind of fish is that? Can we zoom in on that? And
Yeah,
why does it
I
appear to
thought
be
was
swimming
more the
backwards?
same, but
It might be. I haven't been around for all the fish, so if you think we've done it before, feel free not to.
We'll come in. All right, video. You can zoom.
Zooming in. Holding here. Zooming in. Holding.
So is this a fish that we've seen before on this dive? It's another epigonus.
Yeah,
We can
this is
keep moving.
another epigonid,
Great, thank you.
probably in the genus Epigonus. We've seen them quite a bit, actually.
Great, we can move on then.
Great.
There's more of the purple coral just upslope of the
I saw the
fish,
other purple
and
coral.
it looked to me as if
Full wide.
possibly the polyps are open on one of them.
Do we want to go and look at that, or do we feel fairly confident that the other one was a Victorgorgia too? There's a sea star in the middle.
Both the same coral. The two purple things in the middle of the screen.
Well, not sure.
Is there interest here?
Yeah, the lower one looks like it's a little bit lighter in color.
So I need to know whether we want to zoom in on this or not, or whether we need to keep moving. Or if
I think we could keep moving because the only purple coral that anybody knows is the Victorgorgia.
Okay, great.
There's a fluffy coral coming up in the upper right that we haven't seen yet.
Can we zoom on the fluffy
It's
coral,
a
please,
plainer,
Pilot?
very fluffy, dense coral.
Yeah, I think it's that light pink one that's sort of sticking out among the sponges.
Absolutely.
Thank you.
Possibly. Okay, slow it down. You can go in video.
Okay, zooming in.
To do most of these shots flying just because of all the
Holding.
Oh, I think there are some shots being fired in the event log. There are people saying that there are actually other corals that have the purple color. May not be the only way to ID that particular coral. But what are we looking at now?
Yeah, so Scott's point is that there is another purple octocoral, clavularia, but it doesn't create its own branches. It overgrows stuff. So that's the trick. When you zoom in on those, you try to figure out whether it's overgrowing something or whether it really has its own branches.
Very cool.
Didn't Ogden Nash write a poem about purple coral?
Sorry about bouncing around. I'm just trying to land on this one rock without hitting anything.
Ready to zoom?
I don't know who he is, sorry.
I never saw a purple coral. I never hope to see one.
Start in.
Zooming in.
Is that a joke, Bob?
Yes, Ogden Nash wrote, I never saw a purple cow.
Ah. So is this another primnoid, or is this something different?
Well, I don't think it's a primnoid, but I'm holding off. I think it may be a Chrysogorgia, maybe, but Scott or Asako or Sonia, I think, would be a better to answer it.
So it's not a primnoid. All right, coral people, tell us what you know. Oh, they're saying it's a tough one. They're stumped right now. So maybe it's a Chrysogorgia.
Go ahead.
Oh, I don't know if there's a whole lot of agreement here. It seems like there's like, oh, it could be a Chrysogorgia, but we're not really sure.
Copy that. I think we're actually shifting over in 4 minutes.
Pilot, is there any way to zoom anymore?
I don't know
Crew.
this video anymore. I think this is pretty tight. Those are
Yeah.
really small follow-ups.
Yeah, so I think this is pretty much as much zoom as we have.
Okay, I'll zoom out.
Do you have any more in?
Oh, that's all I had.
Yeah, okay.
Sorry, I didn't catch what you just said.
That's all right.
Zooming out. Full
yeah,
wide.
so pilot, I actually adjusted heading. It looks like we had a slight terrain shift, so now I'm about 10 degrees.
Cool.
Actually,
just hang out for 1 second.
Can we zoom
Yeah.
lower on that
We're gonna
colony?
swap
Try,
video
I believe, towards the base.
Towards the base.
Is that a possibility?
Yes.
Okay,
Just
great,
looking
thank you.
for it. And video swapping out. So it's going to be just a minute.
Yep.
Maybe.
So we will be more than happy to
Maybe Nolan will
zoom
sit
at the
down.
base. We just need a moment because there's a watch
for the
change
base
at the video
roll.
station.
Copy that.
Sorry to rush you. I know you're doing your handoff, but
I think they're starting interviews here or something like that in 4 minutes.
Yeah, they're gonna, but we'll be out of here.
So I think we've managed to zoom in on the base of this peachy coral. Oh, look, more little purple dudes.
That's the other purple dudes.
Is that the other purple dude? Or is that the same purple dude?
It's not like DeGorgia, that's the other guy. Anyway, sorry.
All right, so coral people.
There's the hold fast.
Well, I'm going to maintain that it's a Chrysogorgia, and it could even be something close to Chrysogorgia crises. But I think that's the best we're going to be able to go to right now.
What's the purple stuff at the base of this?
Could we zoom on the purple things, which I believe I just called purple dudes earlier?
That's all the zoom we've got.
That's all the zoom we have. So we were looking at the holdfast of that coral. So are the purple ones the stoloniferans?
Must be.
Yes,
And
they
there
are.
you have some open.
Yeah, pilot. As you're zoomed in, it actually looks like we're reaching the top here. Then we might go.
Is that a little drop?
Yeah.
So this is the other purple one that Scott was suggesting that we have to be careful about. So this is the one that either grows on a rock. Well, I mean, there are other stoloniferans, but they don't create their own skeleton. So this is one of the ones that doesn't create its own skeleton. So that's the tricky thing with these purple things.
Now I'm actually pointed north.
Thank you, Chris and Scott and everybody else who's in the event log. I think this is a really interesting conversation.
So if you want to pull ahead and see.
All right, so I think we're going to continue along. Please shout out when you'd like to look at something. As a reminder, if you look in the Sirius feed on stream 2, you will see just how much life there is. This is a super, I mean, Chris, correct me if I'm wrong, this looks to me like very high density of both sponge and coral.
You bet. This is what I consider to be a high-density community.
So Shirley Pomponi is one of our other sponge experts, and she's not on the line right now. And I can tell you she's going to be so jealous that she missed out on this field of tubular sponges.
And it's just like really blowing now.
Yeah, but it's just rushing down that thing on the other side.
It's back down here. Sorry.
So what else might you be interested in looking at?
Because if you look on your sonar, you definitely got something in the back there.
Yeah, we've got a little bump here and then something off towards the north. Yeah. So we can actually get the ship sort of moseyed in a little closer towards the north, and then maybe it's just that we're looking at.
All right, so we're just moving along here, looking at these just fantastic fields of sponges and corals. I don't know what we expected to see today, but this is pretty awesome. And I just love this view as you can look back and see the ridge continuing with just tons and tons of biology. All right, so we're just having a quick ROV watch change. It's totally awesome. Yes, so Diva just walked back in the room. For those of you that are following along, there's two of us, a biology lead and a geology lead. I'm Deb, I'm your geology lead, and yet I have been managing to semi-competently talk about corals and sponges with a lot of help from our shore-based science team. But now Diva's back and very jealous that she missed the fields of sponges.
Was that just drifting, pilot?
What's that?
Was that just drifting?
You absolutely can, but I just wrote it into the event.
You read me okay?
Yep.
So how is it compared to yesterday?
I don't believe
not
so.
nearly as bad.
We haven't collected anything
Zoom
since
here.
I have been here. So if you collected something, maybe, but...
Yep.
So here are several more fish. I think we've seen both of these fish before, the Epigonus,
Yes.
and is that the roughy again that we saw? Yeah. So we've actually seen both of these fish. And it's been very interesting. Diva, you missed it. We saw two different types of purple coral. We saw the Victor Gorgia that we've seen before, and then we saw Stolonifera. It's a different type of purple coral.
I just had visions of coming back downstairs after having been upstairs filming, and you telling me that you'd seen, I don't know, you'd found like a whale fall or something. Coming back downstairs after having been upstairs filming, and you telling me that you'd seen, I don't know, you'd found like a whale fall or something.
You can keep hoping for that whale fall.
Yeah, I think it's bridge.
Okay.
Okay.
Do you hear that video?
All right, science team.
Seems
this is
like
Kelly
bridge
here
is
on board
bleeding
the ship.
through. I
I want to let you know
hear
that
Diva
right now,
over
Deb and
bridge.
Diva are actually stepping away from their role as your onboard science lead here for about the next 45 minutes. So Brian Kennedy, here on board the ship, whom you all know is going to step in. He'll be your onboard
We're not
point
listening
of
to
contact
EVNT.
while we do a live interaction with about 100 high school kids
coming
here
over
in
bridge.
Saipan. So just continue talking to him. Making
come on out,
comments in the
video.
event log like you typically do, making requests for close-ups during this
So where
time.
are
We've
we,
already
heading
relayed
here
to
now?
our pilots that we would like to get as many
Heading
great
to
close-ups as we can
waypoint
to share
2,
these incredible environments
roughly
with the kids
31
back home.
degrees. I can get a more accurate heading here, but
Did
looks like
everyone
we're going
copy that?
uphill.
Yeah, local high looks like due north right now. A little bit west to north.
So this is waypoint 2 up here.
University of Hawaii,
Go
did
over
you
the
copy?
bump and then continue up to get to waypoint 2, 22 degrees.
Thanks, Patty.
So do we want to head straight there, or do we want to go along the local high?
We're going to follow the local high.
Follow local high.
We're going to follow this ridge.
Yeah, so that looks like due north to me.
Well, you got to look out ahead. So you see this ridge line here?
Yep.
Looks like it's,
Yeah, it's right in front of me.
yeah, but it's going up to the, more to my starboard.
Okay.
Well, good morning for those of you in Asia, and good afternoon or evening for those of you in North America. This is Brian Kennedy. I'm a marine biologist
so
and RV navigator with the Global
350,
Foundation
something
for
like
Ocean
that?
Exploration, and
Well,
I'll be
we're gonna
filling
move
in
you
for
closer to the
Diva
ridge a little
and
bit, I
Deb
think.
for the
Let
next
me
30,
push out
45 minutes.
Before we do that, and then we'll
So
adjust
right
from
now
there.
we are just figuring
Copy.
out the ridge looks like it's changed
So
directions
while
a
we're
little
here, can you
so our RV team is
spin up
just
hydraulics
setting
and,
up a new
deploy my
move
lower swing
to stick
arms?
with the
You don't need a lot.
the local high,
hydraulics.
and then they're going to actually deploy
Let's get this
some of
fish in
the
the
extra
distance
lighting on
video.
D2 on our swing arms. So we'll be here just a minute, and while we're doing that, we're going to catch a
Okay,
couple zooms.
lower port.
Looks like another one of the Epigonus deepwater
I
cardinal fish
go
we've been seeing all
left
morning in great abundance.
first to make sure it's stowed.
It's kind of interesting, the comparison from today's dive from yesterday's dive. Yesterday's dive we saw some of the bigger predatory fish, but not as many small prey fish. On
Now,
today's dive,
deploying.
we've seen almost
Yeah,
all
make
of
sure
these
you have an
smaller
eye on it.
Epigonus,
Yeah, I'm looking at the
and only
port
a
rail
couple
cam.
larger roughies. We haven't seen really anything other than roughies that would be like predatory.
Hey, come on
Okay,
out, video.
shoreside, this is Okeanos video.
That's
Just
good.
want to let shoreside know that I'm going to be changing stream 2
Up a little bit.
to be a live camera
like
from the
that,
control
please.
room shortly. So if there's anything special you want on stream
That's fine.
3
Okay.
quad split,
do the
please
same
let me
start.
know. At the moment, I'm going to change the first feed of the quad split to be the Sirius camera, so you can continue to have that second
right.
feed.
Yes.
Watching my rail cam, and I'm deploying. Match.
That's good. Thanks.
Yep.
Otherwise, it reflects off the arms.
Gotcha.
So, I mean, if you were to leave them stowed, you might as well turn them off.
Okay.
You know what I mean?
Yep. Are the uppers good while we're spun up?
The uppers are fine.
All right.
Yep.
Gonna spin down.
And with like the swing arms and stuff, you really don't need to ramp up because they don't really use any flow.
Okay.
Like you still have good pressure at the low RPMs. You just don't have any. You limit your max flow.
Okay.
So for like the craft, you want to ramp it way up because it uses pretty much all the flow that we have. Alright, so we can probably move like 15 meters at his current heading.
Due north?
Yep.
Well, everyone on shore, well, I'm going to take this opportunity while we're getting the RV in position for a couple more zooms and to continue up
Going
this
up.
ridge to say, hey, Mom, happy Mother's Day.
Bridge RV nav.
I know I haven't called yet today, but because it's Mother's Day, all
So
the
I'm
satellite
getting
phones on the
a
ship
little bit of heading
lines are
jerks.
quite busy
That's
with
tether,
everyone
right?
calling home. So I figured I'd take this
Where's that
opportunity to
current?
say hello and thank you for being my
usually,
mom. And I'll
part
call
of
you
the
later
feed.
today when I get off watch.
You're talking about left and right?
Yeah.
Like you're gonna
So
get
this
a
has
little
been,
bit just because
I
of
would
the
say,
current
one of the highest
picking up.
densities of sponges I've ever seen.
We're ready
Chris,
for a
have you
15
ever
meter
come
ship
across
move.
places like this in the Hawaiian
Bearing
Islands with this
000
many sponges?
due north.
I've
at a
certainly
speed
seen
of
areas
0.2
of super
knots.
dense corals with
pretty
sponges intermixed, but this being a
See how it
sponge-dominated
goes up
community
to
with
my
the corals
server.
kind of
Thank you,
intermixed
Rich.
is new for me, at least. Pilots, are you set up to start doing zooms on things again?
Hey, come on in video. Yep. Yeah, we're ready. No, hold that. I'm just looking for see what's out here. The big guy? Yeah. I'm about out of range on that guy. Waiting on the ship move. Should be there in a moment.
All right, Chris, we'll get there in just a minute. We're waiting for Sirius to catch up.
This guy?
Yeah, I think that's it. Yep. But in the meantime, until then, can we go in on one of the purple corals? One of the little,
Yeah,
what we think might be a Victorgorgia. All right, Asako, we're going to head back towards the Victorgorgia right now. We're waiting to get enough leash to get after that large reddish-brown colony that Chris wants to see.
Right, come on in.
Looks like there might be, oh wait, I guess that's a ctenophore or something. I can't tell what that is in the water column floating around. Maybe it's quite quick.
All right, come on in video.
All right, so we're just going to take the zoom on this purple colony that
I'm gonna
we're
get in
thinking is a
little closer,
Victorgorgia.
hold it.
We got a really good look at one earlier with all the polyps closed. So hopefully this one will have a few polyps open to get a better look at it.
Yes. Just keep
So
talking,
we're right
go
on
through
this
it.
really
I'm
steep
listening
ridge
to you,
back in this high
pilot.
density sponge and coral colony. vehicle's currently at 455 meters on Esmeralda Bank, which is just west of
on
Tinian
EVMT
and Saipan. In the,
you won't.
In the Commonwealth of the Northern Marianas.
It's on event.
Looks like we've also got a little crustacean to the left of the coral. Can't quite tell what it is yet.
Okay, I think I'm moving now.